mitosis. So next one is the differences between mitosis and meiosis. So generally mitosis, it takes place in the body cell, that is the vegetative cells, whereas the meiosis, which takes place in the special cells of the sex organ. So here in mitosis, here the 2 daughter cells are formed, whereas in meiosis 4 daughter cells are formed. So here in mitosis, the daughter cells are identical to each other and to the parent cell. Whereas in meiosis, the daughter cells are not identical to each other and to the parent cells also. So here in mitosis, the number of chromosomes remains constant. Whereas in meiosis, the number of chromosomes become half. Okay, so next one is that the functions include the cellular reproduction and general growth and repair of the body. So this mitosis mainly helps in, that is mainly for the cellular reproduction and general growth and repair of the body. So whereas the meiosis, the function which includes the producing the organisms having the different characteristics through the sexual reproduction. So these are the differences between mitosis and meiosis. So next one is the significance of mitosis and meiosis. So significance of mitosis is, so mitosis is important for the sexual reproduction indirectly, and it allows the sexually producing organisms to grow and develop from the single cell into a sexually mature individuals. So this allows organisms to continue to reproduce through the generations. So asexual reproduction, so here asexual reproduction, it is the process by which a single organism produces genetically identical offspring. So this type of reproduction, which is most common in the single cell organisms like bacteria, but it can be found in more complex organisms as well. So next one is a meiosis, significance of meiosis, which provides diversity. So one of the benefit of sexual reproduction is the diversity it produces within a population. So that variety, it is a direct product of the meiosis. So every sex cell which is made from this meiosis has a unique combination of chromosomes, and this means that no 2 sperms or egg cells are genetically identical. So every fertilization event produces new combinations of traits. So this is why siblings share the DNA with parents and each other, but they are not identical to one another due to this meiosis. So recombination at meiosis plays an important role in the repair of the genetic defects also. This recombinations which occurring in the meiosis, so which also plays important role in the repair of the genetic defects. So conventional view that it generates by the recombination and sexual reproduction the genetic diversity on which the natural selection can act. So it is essential, at least in animals, for the reprogramming of gametes, which gives rise to fertilized egg. So this is about the importance, significance of this mitosis and meiosis. So here some of the terms which are used in this cell division are okay, diploid. So here the process of, so generally meiosis, it is a process of reduction division. So in which the number of chromosomes per cell is cut into half through the separation of the homologous chromosome in a diploid cell. So diploid means 2 sets of chromosomes. So it is the term which is used to refer a cell that contains both sets of homologous chromosome. Diploid means 2 sets of chromosomes. Haploid means 1 set of chromosomes, and it is a term which is used to refer a cell that contains only a single set of chromosomes. Next, homologous chromosomes. So homologous means the term which is used to refer the chromosomes that each have a corresponding chromosome from the opposite sex parent. And homologous chromosomes are similar in shape, position and information, but not identical to each other. So they are similar in shape and position, but they are not identical to each other. So here, for example, homologous chromosome, a homologous pair of chromosome, that is paternal and maternal. So here the replication. So homologous pair of chromosomes doubling, that is sister chromatids of parental chromosomes. So important definitions of this meiosis is it is a process of reduction division in which the number of chromosomes per cell is cut into half through the separation of the homologous chromosome in a diploid cell. So here, generally in the cell which contains four that is two sister chromatids. Okay. So here finally only. So here when we observe this, so again here another difference is also there. At the end of the meiosis, the individual gamete cell has divided from one cell to four. So generally males produces four viable sperms, but females produce the one viable egg and three non-functional polar bodies. Okay, so this point also important. So here this one, so chromosome after S phase. So here the chromosomes at the beginning of the mitosis and here the after mitosis. So two equal. Okay, so here after meiosis, so for example, here the two chromosomes, that is capital B chromosomes and small b, two sets of chromosomes. So at the after mitosis, here the two parental, so so the two daughter cells, they belongs to the they are similar to the parent, but here in meiosis, so half only, so they are not resemble the parents, so only half character only. Okay. So here the four haploid set of chromosomes are produced in the meiosis. So tetrad, so it is the structure which containing four chromatids that forms the during the meiosis. So four chromatids present at the and the chromosomes, it is called as a tetrad. So crossing over, the process in which the homologous chromosomes exchange the portions of their chromatids during the meiosis. So here the exchange of the chromosome will be taking place. So these are the two homologous chromosomes. So formation of tetrads while going through the process of crossing over. So this occurs during the prophase 1 of the meiosis, crossing over. So meiosis have mainly two divisions, that is meiosis 1 and meiosis 2. So prophase 1, which occupies 90% of the meiosis, and chromosomes begin to condense and homologous chromosomes loosely pair along their length and precisely aligned gene by gene. So in crossing over, the DNA molecules rejoin each other. So this is a prophase 1, that is here the formation of thin thread-like structure will be formed. So each chromosome pair is visible as a tetrad, that is four chromatids. So each tetrad has one or more chiasmata. Regions where the crossing over is occurred, it is called as a chiasmata, and hold the homologs together until the anaphase. So centromere movement, spindle microtubule formation, nuclear envelope breakdown, and nucleoli dispersal occur as in mitosis. So in late prophase 1, that is kinetochores of each homolog attached to the microtubule from one pole or other. So then homologous pairs move towards the metaphase plate in metaphase 1. So here the pair of homologous chromosomes are arranged on the metaphase plate with one chromosome of each pair facing each pole. So both chromatids of the homologs are attached to the kinetochore and microtubule from one pole. So those of the other homologs are attached to the microtubule from the opposite pole. So here this one is a metaplate, equatorial. So metaplate formation. Okay, they are attached at the equator. So in anaphase 1, chromosomes move towards the poles and sister chromatids remain attached at centromere and move as a single unit towards the same pole. Homologous chromosomes, each composed of two sister chromatids, and move towards the opposite poles. So first tetrads are present, but here only two chromatids will be separated here. Okay. So in telophase 1, at beginning of the telophase, each half of the cell has a complete haploid set of chromosomes, but each chromosome is still composed of two sister chromatids. So in meiosis 2, no interface, and similar to the mitosis, cells have one set of sister chromatids and nuclear membrane breaks down, centrioles separate, and each of the daughter cell forms a double-stranded chromosomes and moves towards the equator. So sister chromatids line up in metaphase 2, they line up at the middle of the cell. And in anaphase 2, the centromere of the sister chromatids finally separate, and this sister chromatids of each pair moves towards the opposite poles. So now it's individual chromosomes. So in telophase 2, cells start to split in two, and each cell has one set of chromosomes. It is called as the haploid. And nucleoli form opposite poles of the cell, and cytokinesis occurs. And after completion of this cytokinesis, there are four daughter cells. All are haploid. So here four daughter cells are produced after meiosis. Okay, so this is about the meiosis 1. So in interphase, so here meiosis 1, that is the tetrads formation. So in metaphase 2, here they are attached at this metaphase plate, and they are separated. And here the tetrads to diads, two with chromosome with two chromatids will be separated here. So each containing the two chromatids. Okay. So in meiosis 2, so this two chromatids again same, it is similar to the mitosis. So here the first one is a nucleoli, and a nuclear membrane will be disappears, and here they in metaphase 2 again here they are attached at the metaphase plate equator, and they are separated, and each contain the haploid set of chromosome. So here the non-disjunction. So it is the failure of the homologous chromosomes of sister chromatids to separate during the meiosis. It is called as a non-disjunction. So non-disjunction which result with the production of zygotes with abnormal chromosome number. So remember, so an abnormal chromosome number, that is the abnormal amount of the DNA. It is the damaging to the offspring. So this disjunction means it is the failure of the homologous chromosomes to separate during the meiosis. It is called as a disjunction. So due to this disjunction, so many diseases also occurring. So impact of this non-disjunction, the first it is called as the monosomy. So the second it is called as a trisomy. So in one, there is only one one chromosome is absent. So in another, extra chromosome will be present. So if an organism has trisomy, that is 18, it has a 3 chromosomes in the 18th set. So the trisomy 21, 3 chromosomes in the 21st set of chromosomes. If the organism has a monosomy, that is 23, it has only 1 chromosome in the 23rd set. So here due to this non-disjunction, so some diseases also occurs. So Down's syndrome. So it is a trisomy of 21, extra chromosome in the 21 set of chromosome. Turner syndrome, that is the monosomy of 23. That is one chromosome is absent here. And Klinefelter syndrome, that is trisomy of 23. And Edwards syndrome, that is trisomy of 18. So this is due to this non-disjunction. Due to that is they are not separated. Homologous chromosomes are not separated. So due to which, so some syndromes will occur. So it is harmful to the individuals. So this is about the meiosis and the real significance and differences between mitosis and meiosis. Okay, sir.
Any doubt? Any doubt, my dear friends? Nafis, Sultana, Manasa, Vijay. Vijay from which college, Amma? Okay. Madam, thank you very much. Really, it is more important, most important topic. And also importance, the differences between the mitosis and meiosis, and important points you have covered in the meiotic cell division. That is also we can call as reduction cell division. It is mainly we are observed in the gametes. Gametes are so, my dear friends, this is the most important topic, and tomorrow, Madam will going to explain about the molecular biology, especially DNA and RNA structures, and the transcription translation in the process of protein synthesis. Madam is very exclusively expert in the molecular biology also. So tomorrow we will meet in the same time. Thank you very much, Madam.
Thank you, sir.